Hi, good evening. My name is Gabriela Sanchez. Today is Tuesday, November 17th, 2020, and I'm working on week 3's lab 5, part 1 for bacterial morphology and staining techniques. I'm going to be working on the first lab experiment, or the first part of it rather. And I have my materials already ready. I have 2 slides prepared. One is the direct M, and I have the circle here already drawn on there. 1 means it's sample number 1. Direct means that it's going to be a direct stain, and it's going to be obtained from the mouth. So that's why it's the letter M. On the second slide, I have 2 negative M. 2 means it's the second sample. Negative, it's going to be a negative stain. M, it's going to be a source of the mouth. I have an extra slide because I'm going to be doing the staining with the Congo red stain. So for the 1 direct M, what I'm going to do, since I already have my slide made up, I'm going to obtain one of the sterile swabs that's packaged. I'm gonna vigorously swab the inside of my cheek as well as the gum line. I'm going to smear the swab sample onto the slide, and once I've done that, I'm going to let it air dry, and I'm gonna put a drop of the crystal light, and I have it here, the crystal violet gram stain. I'm gonna place 1 drop on there once my sample has dried. So it has to be dry first. Make sure that it's dry. I'm gonna apply the sample or the gram stain rather onto the slide. Once I've done that, I'm going to get the staining tray, and I'm gonna pour a little bit of water just to kind of let that excess staining roll off the slide. And if there's excess water, I'm gonna just dab it with a paper towel, not smear it. I'm gonna dab it just to get all the excess water or solution away from there, and let it sit and dry. And then I'm gonna look at it in the microscope, and I'll be taking pictures and uploading them to the HOL lab. For the second one, the second sample, negative sample, I'm going to, first I'm going to pop this open, the Congo red. I'm gonna cut it open. I'm gonna apply 1 drop onto the slide. I'm gonna then grab the second swab from here, vigorously swab the inside of my cheek as well as the gum line, and I'm going to dab that onto the stain, the Congo red stain that's gonna be on the slide. I'm gonna try and get as many cells off of the swab onto the slide. Once I've done that, I'm going to do a smear with a blank, I don't know if you could see it, with a blank slide. Once I do that, I'm going to not put it right on top of it. I'm gonna smear it at an angle. So from the top all the way down so that it could smear all the way to the bottom. I'm gonna let that sit and dry for a few minutes. I forgot to mention on the previous one, on the direct M slide, I'm gonna have to put this on, sorry, I'm going back and forth. With the paper, with the clothespin, I'm gonna heat this before I apply the crystal violet stain. And then I'm gonna put the stain on there. So I failed to mention that on the first part, sorry. But once I smear the second one, coming back to this, I'm gonna let this sit and look at it under the microscope, take pictures and upload them to my HOL lab. And then I'll do my second part to see what I came up with. I also have to do the second part of the exercise with the broths, the E. coli, the S. cerevisiae, and the S. epidermidis. Not sure if that's supposed to be included in the second video or not, but I'll try and incorporate it as much as possible so that you know that I'm completing both of parts of the exercises. So that concludes my video, part 1 for week 3, bacterial morphology and staining techniques.